If you are familiar with Madagascar movie, you may know King Julian. King Julian is a species of lemur called the ring-tailed lemurs, and lemurs are only found in natural habitat in Madagascar, which is an island situated in the southeast of the mainland Africa. But I'm not here to talk about King Julian, but another species of lemur called the black and white ruffed lemurs. Hi everyone, my name is Eliette Noromalala, and I'm a graduate student from primate behavior and ecology. And today I'm going to talk about the factor influencing intestinal parasite in the black and white ruffed lemurs. The black and white ruffed lemurs are found in the eastern part of Madagascar. So the green dots and the red dots represent two populations that are genetically related. The lemur is critically endangered species because of deforestation and human activity. And they are arboreal because they spend most of their time on the tree as seen on this picture. And they are also frugivores because fruit makes up a higher percentage of their diet. Fun facts about them are they participate in seed dispersal because eating fruit, they disperse their seed and reconstruct their ecology. And they are also the largest pollinator in the island of Madagascar. To study them, I went to Ramafan National Park, which is situated in the southeastern part of the island. And Manguev Ramafan is my study site, which is in the southeastern part of the Ramafan National Park as well. So the area covers the forest covers an area of 435 kilometers square. It's a pristine rainforest with a mid elevation between 600 to 1200 meters. I studied two communities of the black and white ruffed lemurs seen with purple and pink in here. And the two communities are slightly overlapping. I went there and collected data. So one of them is behavioral data. So every 5 minutes, I recorded everything that the lemurs did and extract the feeding data from overall data. I also collected climate data, which is temperature and rainfall of the habitat. And also I collected fecal samples, stored them in 10% formalin for further analysis. And all of the individual followed were radiocolored and tagged because I was part of ongoing project. Why? Because I want to describe the parasite pattern, the number, and the species diversity. And I also want to estimate the monthly percentage of fruit consumed by the black and white ruffed lemurs. This is because I want to see the relationship between diet and parasite and the relationship between climate and parasites. So for the feeding data, if the lemurs eat leaves, fruits, flowers, and buds, I calculated the percentage of the fruit that they eat. And for parasite isolation, I used morphological identification and used two techniques. First, flotation. So flotation was used to target species of parasites that have a light-specific gravity. So I used sugar solution and all the parasites float on top, and I put cover slip on that and observe it on the microscope. And I also used sedimentation for parasites that have a heavy specific gravity, and all of the parasites sink at the bottom of the tube, and I take some of that and observe on microscope. And what you see on microscope is thin like this. So I recorded the type of the parasite, the species, and the number because I counted them. I have two hypotheses. So first, when they consume more fruit, their parasites should decrease, so the infection and number and diversity of parasites. Why? Because when they access to good food, adequate food like the fruit, their parasite, they boost their immune system and cope parasite. And my second hypothesis was when the temperature and rainfall increases, the parasite infection number and diversity should increase as well. Why? Because wet and warm condition is really good for parasites to hatch eggs and to increase their reproduction. So what did I find out? I want to point out first that this is the first investigation in intestinal parasite of the black and white ruffed lemurs in wild population. So we found 3 species of protozoan, first Balantidium, Emeria, and Entamoeba. And we also found 2 species of nematodes, which are Callistura voseli and Strongyloids. We could not identify these eggs, and we only could identify larvae at phylum level, and we found larval nematode. The question is there a difference between difference in parasite prevalence across month of both sex, male and female? The answer is yes, because Balantidium followed by Callistura have higher prevalence, and Emeria followed by Entamoeba have lower prevalence. And Balantidium have higher prevalence in March, and Callistura voseli have higher prevalence in May. And if you see at the 2 species of nematodes, Callistura and Strongyloid, there is a pattern of an increasing trend that February have lower prevalence and May have higher prevalence. Another question is that, is there a difference of parasite richness and diversity across month for all the individuals observed? The answer is yes as well, that February have low species richness and March have high species richness. And if you look at the diversity, you can see that April, not March, have high specific species diversity, and February have low species diversity. One more question. Is there a difference in parasite species diversity for species of parasite across month? The answer is yes as well, that Balantidium followed by Callistura have higher species diversity, and Strongyloid followed by Emeria have low species diversity. When we compare the difference across month, statistically, if they are significant, food consumption and minimum and maximum temperature and rainfall, those are higher in March compared to May. And all of the parasite data, such as Callistura voseli, Strongyloid, and larval nematode count, they were lower in March compared to May. What all of that means? That means that when they increase fruit consumption and temperature, they decrease larval nematode count, and when they increase temperature, they decrease Callistura egg count. The take-home message is, because we only find 5 species of parasite, and that's common in endangered species such as the black and white ruffed lemurs. And it's not surprising to see high prevalence in Balantidium in frugivores, because fruit contain a tiny amount of compoundary compounds that have antiparasitic aspect. And also the accessing for adequate food boosts their immune system to cope parasite. And what is next is we are collecting from over a year data using molecular methods, and we send Callistura voseli to be shipped to be sequenced to do nested PCR for not only Callistura, but over species of nematode as well. Finally, I would like to thank my committee members and collaborators and all of the institution, the funding and assistant and lab. And this is the reference I extracted my information for this presentation. And thank you for your attention. Any questions? Yes. Yes, the trees, the plant trees, I know the Malagasy name of that tree in the rainforest. They are the main pollinator of the trees in the rainforest in the east part of Madagascar. Yes. Yeah. Okay. Yes. There is no difference between female and male infections when we did the study. No. Thank you. Okay.
Hi, thank you. So in my project, in this study, we show that we can in fact use HepG2 cells as a model for non-alcoholic fatty liver disease solely with excess sugar instead of excess sugar with excess fat. But first, why should we even study liver disease? In this table, it compares different ages, genders, and ethnicities of people in the United States with non-alcoholic fatty liver disease. And you can see that while there's trends with there being more non-alcoholic fatty liver disease patients who are a little bit older or possibly more obese patients have non-alcoholic fatty liver disease, it does seem to be a trend that this is something that can affect anyone. This is something that anyone can have. More specifically, whenever we compare this across the entire American population, 1 in 3 of US citizens are having this disease. But this is not just a United States problem. This is an increasing problem globally. This map above me shows the amount of NAFLD patients with diabetes in particular globally. And this is an important thing to specify because liver disease is going to lead to heart disease. It's going to lead to insulin resistance and obesity. So these are important factors to remember when we talk about non-alcoholic fatty liver disease. But what is it? Excess sugars in the diet synthesize fats. And whenever the liver is making too many fats or lipids, or de novo lipogenesis is occurring, then this probes the issue of hepatic steatosis, or excess storage of fats. And this becomes pathologic and induces lipotoxicity. And liver disease leads to serious complications that are irreversible. It leads to cirrhosis and hepatic liver cancer. But first, what does it look like? The phenotype of someone with liver disease has a liver with a high fat concentration, fibrosis, glucose intolerance, and elevated fructose 1P levels and reduced beta oxidation. Specifically, it's really important to remember that this leads to irreversible consequences. It leads to cirrhosis and liver cancer, where at this point, you need a new liver. So my study focuses on the dangerous precursors to cirrhosis and liver cancer, particularly steatosis and NASH, which is non-alcoholic steatohepatitis. Particularly in my study, I look at the fat content with oil red O assays and reduced beta oxidation with WST assays. And before any of this was done, I compared the concentrations of cell seeding with crystal violet assays. An important thing to remember before we get started is that excess dietary sugars lead to increased levels of growth hormones in fatty liver disease. Seen here are my HepG2 cells under a microscope. So first, I ask the question, do excess sugars alone drive cell growth or cause cell death? Crystal violet assays were done in order to count the amount of cells, to measure the number of cells. Here, we see that excess sugars aren't really changing the rate of growth. We see, did it show? Yes. Okay. I show that 3 different levels of hexoses. So physiological glucose at 5 millimolar, excess glucose at 25 millimolar, and excess fructose with 25 millimolar fructose and 5 millimolar glucose without growth factors don't have a significant change in the rate of growth, which is all normalized back to the day 1 seeding concentration. But what happens when we add growth factors? More crystal violet assays were done to answer this question. And as you can see here, again, we have the 3 hexoses. However, they're in all growth factors. They have FBS, insulin, and TNF alpha, which again, doesn't show a change in the rate of growth. So what do we have right now? We have excess glucose and excess fructose having the same amount of cells. So what's the difference? What's the problem? So then I ask, do excess sugars increase intracellular lipid accumulation? Is there a difference in lipid accumulation between fructose and glucose? Oil red O assays measure lipid accumulation. So this is how we're going to test it. Here, we have the 3 hexoses with 4 different levels of growth factors. No growth factor, FBS and insulin, FBS and TNF alpha, and FBS and insulin and TNF alpha. All normalized back to the no growth factor. Here you can see that there is an increase slightly with the insulin, which makes sense. However, let's look a little bit closer to the 2 larger groups on the side. Here, you can see that the TNF alpha groups are increasing lipid storage with excess sugar, whether or not insulin's included. So why is that happening? Here, we see that the amount is the same for fats and fructose, but you just saw a difference in the last slide. What's happening is even though there's an increase with TNF alpha, it's the same for fructose and glucose. So again, we have the same amount of cells and the same amount of fat. What's happening? So now I ask the question, do excess sugars increase mitochondrial function? Do different growth factors increase mitochondrial function? This was tested with a WST8 assay, which measures mitochondrial dehydrogenase enzyme activity. Excess fructose here shows lower mitochondrial activity, even though all glucose is showing twice as much mitochondrial activity, except in one group, the TNF alpha group. So why is that happening? Does TNF alpha signaling interfere with the mitochondrial function? How are these results connected? So with fructose, we have a reduced mitochondrial function. With glucose, we're having twice as much mitochondrial function. But why? Increased TNF alpha increases gene expression of fatty acid synthase and acetyl-CoA carboxylase, which we show in the oil red O assays have more lipids with TNF alpha, which follows. Then the WST assay shows reduced mitochondrial activity. A decrease in healthy mitochondria also follows. Lastly, we show that with excess fructose, there's more TNF alpha, which then signals the stress in lipogenic pathways, which creates more trans factors, which are creating the uncoupling protein UCP4, which with extra expression is then creating more reactive oxygen species, which can be measured in a glutathione assay. So what's next? Glutathione assays to measure the redox balance. Thank you. and all of my references. Questions? Yes. That's actually really interesting, and I could barely hear you, so just to make sure I have the question right, you're asking why I used HepG2 cells for my model? Okay, so in literature, and the references showed in the last slide, there's a lot of rat and human models that show the exact same things that I was showing with my assays. We have increased lipid accumulation, decreased mitochondrial function, and what my presentation is really showing is that we can use HepG2 cells. It's really not inferred that we can use HepG2 cells, but at the end of it, it's saying, yes, HepG2 cells are a valid model, and we can continue to compare these to perhaps Aldose the knockout cells, which other people in my lab are working on. And this can go forward as a good control and can be a model for non-alcoholic fatty liver disease. Yes? So first, we have these flasks that I grew stock cells in, right? And so they grow up to a certain point where I believe I showed the HepG2 cells underneath the microscope, right? And in this, then they become trypsinized, so they get lifted off the plate with a series of chemicals and incubated and resuspended in media and plated across the 96 wool plates you see here. Then stains are added, and they, depending on the assay, for example, crystal violet assays measure the number of cells. So it's staining the cells so that it can be measured in a spectrometer in order with the wavelength. It shows a quantitative amount of cells rather than just looking at it and saying, well, that is darker. Does that answer your question? So here, for example. Okay, yeah. Okay, so all of these are normalized because whenever they do get plated in a 96 wool plate, you want to have the same amount of cells in each plate to have an appropriate measurement of whether or not there's more cells in the plate or more lipids in the plate or more mitochondria being used. And if it's not normalized back to the concentration, like a singular 1 concentration, you don't know if that's just me messing up a pipette or if that's actually scientifically what's happening.
Hello. As mentioned before, my name is Cade Walker, and today I'm presenting on the analysis of wildlife crossing structures along the I-90 corridor. So as global transportation and infrastructure expands at a rapid rate, global biodiversity is threatened by the barriers that roadways make, preventing habitats to be connected, fracturing them, reducing population genetic variation, as well as preventing populations from interacting with each other, isolating them, and ultimately reducing the biodiversity and ultimately reducing the productivity of our ecosystems in the area. And as you can see on this picture on the left, there is a great example of how roadways fracture our habitats. They separate these beautiful areas that wildlife species can use to inhabit and increase their populations in. So it is important to evaluate the usage of crossing structures and their characteristics to design effective passages for future projects. This is a focus that has been in practice since wildlife crossing structures were implemented. The article that I cite this from was actually an article done in Spain. So this is a very worldwide solution to how we think of wildlife crossing structures and how we design them. And then as you can see on the left, we have 2 wildlife crossing structure types here in Kittitas County from our very own crossing structures. The top structure there is the wildlife overcrossing that some of you may have seen as driving down I-90 as you're headed towards the west side or as you're headed back towards here. And then the bottom crossing structure is an underpass that is located as well in Kittitas County. And you can see that there are 2 very different types of structures, but they still accomplish the same goal of reconnecting habitats. And then as my third point there is this project, a project was made to construct wildlife crossing structures along I-90 in Kittitas County in 2008. It was in conjunction with Washington State Department of Transportation, United States Forest Service, as well as Washington State Fish and Wildlife. And basically their goal was to reconnect these fractured habitats as well as reducing wildlife and vehicle collision incidents. So going into the Wildlife Crossing Structures project, there was 14 connectivity emphasis areas. As you can see, it begins at milepost 55.1, which is north of Cachilas Lake. And then it ends at milepost 70.3 over by Lake Easton. And you can see each of the connectivity emphasis area, and of those emphasis area, there are 27 crossing sites in them, and 11 of them have already been constructed as of 2021. 4 of them are in the Price Noble connectivity emphasis area, which is where most of the recent structures, as well as that overcrossing structure I showed earlier, is located, as well as 2 in Gold Creek and 2 in Resort Creek connectivity emphasis area. There's also 1 crossing site in Rocky Run, Wolf Creek, and Townsend Creek as of 2021 for the total of the 11 constructed. So there has already been quite a bit of crossing structures already implemented throughout the I-90 as of this year, with further more to come. So crossing structure usage, as you can see off these videos here, that you have quite a number of animals using them. On my right, you can see the elk crossing over the overcrossing on the eastbound I-90, as well as the coyote crossing over on the westbound section of the overcrossing. You can see that there is a variety in not only the number of individuals crossing, but also the species, which is important because they are used by multiple species, but were not designed for use by humans. However, human presence can still be affected, can still be felt on these crossing structures. And I will go into that later. So for my method of my data analysis, I removed the unnecessary data entries after compiling the raw data set that I was given to by the generous people over at Washington State Department of Transportation, or WSDOT. And then that included duplicates, as animals like to move back and forth, as well as removing unused factor levels. So making sure that we are looking and observing levels that were important and of interest to these crossing structures, primarily focusing on such as the structures, whether or not they have been post or pre-construction of whether the data was there or not. So we focused on whether wildlife crossed the structure or not in the terms of successful passage or unsuccessful passage. For a successful passage to occur, the wildlife needs to cross over the structure. If they did not, it's deemed an unsuccessful passage. And basically, we were able to factor that into our data analysis by revaluing the passages, unsuccessful indicating a 0, 1 indicating a successful passage. We then used the logistic regression model of the probability of successful passage as a function of number of individuals crossing, days since human presence, and crossing structure, as well as using any factors of interest that we wanted to observe to see if there's any differences, as well as just looking for significance in these models. So moving into our results, we found that Noble Creek Underpass had a significantly lower probability of successful passage. This was determined using a pairwise function that compared the direction variable of our data set basically which structure was which. And while the underpass had a significantly lower probability, it still had a relatively high number of successful outcomes, somewhere above 90%. And then moving into the figure on the right there, you can see the frequency of successful and unsuccessful passages between those connectivity emphasis areas that I mentioned that have constructed crossing structures. The Noble Creek underpass resides in the Price Noble Creek connectivity emphasis area. And while it has the most crossing structures, it's still relatively small. That may be due to the recent construction, but we'll go into that later. The time period was also found to have significant levels in the success of the probability of the successful outcome, successful passage of wildlife. We found that dawn had a higher probability of successful passage compared to night, as well as you can see on this plot here that night had a higher number in frequency, but not a higher proportion. So understanding how those two different time periods differ in their probabilities is important for further construction and further design. Moving into the number of individuals crossing, which is the population, not the population, the groups that are using these crossing structures, as well as the days since human presence, both significantly increase the probability of successful passage. You can see on this figure on the right here, the probability of the successful passage for the species of white-tailed deer that use the crossing structures as a function of the number of days since human presence. You can see the logistical curve increasing as days after human presence increases. So what our data analysis indicates? The human presence has negatively affected the probability of successful passage, meaning that we need to make sure that we're not negating, but preventing any unnecessary presence on these wildlife crossing structures to ensure that their probability of successful passage stays at high levels at all times, to make sure that we are crossing and reconnecting these areas as much as possible. Moving into larger groups of wildlife and how they are more likely to cross these wildlife crossing structures. As I mentioned earlier, the number of individuals crossing had a significant effect. And as larger groups use these crossing structures, they are found to have a higher success of passage. So taking that into consideration, we need to construct future designs to be wider and longer to accommodate for large individuals, to ensure that that these large groups are continually achieving the probability of successful passage. So no individual crossing structure design fits all. That was mentioned by Clevener and Waltho back in 2003, and that still holds true. But future analysis focusing on different species and their response to the different aspects of wildlife crossing structures can be utilized to help us further design future projects and future constructions. As I mentioned, the I-90 project has 27 proposed crossing structures with only 11 built. With those 16 crossing structures in mind, we need to keep looking at our analysis of these wildlife crossing structures to ensure that we're maintaining the highest probability of successful passage to continually allow these wildlife crossing structures to be effective. I'd like to acknowledge my mentor, Dr. Alison Scoville, Josh Zilstra, and Mark Norman, as well as WASDOT for allowing me to use their data set. Here are my references. I open the floor to questions. So for the species that were found to be crossing and using these crossing structures, the detection method, I definitely did favor the larger species. So understanding how these wildlife crossing structures are utilized by smaller mammals and smaller species is definitely finding how that occurs is something that needs to be accounted for in future analysis, as well as going into how these structures are used by certain species. You can definitely tell in my analysis, I found that it's used a lot by ungulate species. The white-tailed deer and the elk, as I showed in that video earlier, have really been utilizing these crossing structures. And in fact, as it's just almost like they're built for it, because as we all know, when you see a collision occurring on the roadway, you often see deer or elk. So with these wildlife crossing structures, we're definitely seeing a lot less collisions of these ungulate species occurring. So I would definitely say ungulate species are utilizing these wildlife crossings most, but small mammal species and small amphibian species are the ones that are being under.
Thank you. Okay, so Clark's nutcrackers, they are a corvid species, so they're related to crows and ravens, and they have a very wide range across the western United States and parts of Canada. And so in the range map, you can see they range pretty far up and down the coast, including, and so within that range, they eat a lot of different types of materials. So in terms of plant materials and animal materials, they are omnivores. So in terms of plant materials, they'll eat a lot of conifers. And animal materials, they're actually very opportunistic and will mainly specialize in plant material during the winter and fall seasons. In terms of plant materials in Washington, they consume things like whitebark pine, ponderosa pine, and Douglas fir, which I'm sure if you spend much time outdoors in Washington, you'll see some of those. And down in Utah, they're more likely to forage on things like pinyon pine or bristlecone pine. In terms of the animal-based material, I mentioned they're very opportunistic. So maybe like some crows you may see sometimes, they will attack roadkill. They also eat insects and small mammals and will even attack other bird species, like in this image here where they're attacking a tree swallow. But one of their main mutualistic relationships is with whitebark pine. Whitebark pine is a keystone species in high elevation areas and is very important for habitat use. Their seeds are actually very large seeds and provide great nutritional value for nutcrackers among other species, but they are in decline. And the decline is very important because of climate change and fire changes to the fire regime, the mountain pine beetle, as well as white pine blister rust, which was an invasive fungus introduced in the 1920s and has caused a large impact on many white pine species, including whitebark pine. And so what do we do to get away from a declining species is we focus on their survival and reproduction. And so for whitebark pine specifically, Clark's nutcrackers, my study species, are the only dispersal method for whitebark pine seeds to significantly disperse their seeds. And so my objectives for this study were to assess the stability of the nutcracker conifer relationships and assess large-scale temporal and spatial changes in the nutcracker's diet. So to get at this, I had some very broad research questions, including changes in seasons and life stage of Clark's nutcrackers and how their diet changes between those. Their changes in relative diet based on years. I have nutcrackers from 1888 through 2016. So it's a very broad annual scale and also changes between locations. To get at my research questions, I had used museum specimens, and they came from these very nice museums that let me use their specimens. And I actually was able to separate these nutcrackers into groups, juveniles, adults, by life stage, and by seasons across, which are based on previous research dictating when their spring season would be, which is when they normally breed, their summer season, and then their fall, when they typically start catching conifer seeds. From my specimens, I collected emerging feathers. And in this image here, you can see there is a sheath still on the feather. That is what I was looking for in my specimens. They're kind of difficult to find, but I went through each specimen all along their front and back sides and collecting emerging feathers. And these were very important to my research as emerging feathers retain the same isotopic signature during their growth period, and they do not change their isotopic signature after they are done growing. So the emerging feathers will match the collection date in terms of diet. And from these feathers, I ran carbon-13 and nitrogen-15 isotopic analysis to get those, back. And in terms of carbon, carbon does show changes in diet niche, mainly pertaining to photosynthetic origins of the carbon, and nitrogen shows differences based on trophic level. So higher nitrogen would mean they were eating more animal-based materials versus lower nitrogen, where they were eating more like conifers and plant-based material. These feathers were then prepared using a 2-to-1 chloroform-methanol mixture and corrected based on the Seuss effect, which is a baseline change in carbon-13. I'd love to talk about that more after the presentation, but it was a complicated process. So what I got back was this. It has a lot of points everywhere, so it's a bit complicated. But looking at it like this, in the bottom right-hand side, so high carbon, low nitrogen, that would be the diet niche I'd expect them to eat more conifers. And in the high nitrogen, low carbon, so the upper left, are where I expect them to be eating more animal-based materials. And they did become clearly distinct very nicely in this figure. And so my summer adults, summer juveniles, and curiously my fall juveniles were all in the upper range of trophic niche versus the spring adults, the spring juveniles, and the fall adults, which appeared in lower trophic niche, meaning more plant and animal-based materials. Here I did pairwise comparisons to look at them, and summer had significant differences in nitrogen signature, which indicated to me that there were definitely more animal-based materials being consumed during that summer season, with the exception of fall juveniles, which actually overlapped all of my other Nutcracker groups, which was very interesting. In terms of carbon, you see the same pattern, except for life stage is also significantly different in the case of carbon, where juveniles have a significantly lower carbon signature than adults. And so I plotted this over year as well. And so here in this figure, there is a dotted line, and that dotted line represents the introduction of white pine blister rust, which affects a lot of the species that nutcrackers consume conifer-wise. And there doesn't appear to be any changes in terms of nitrogen. So consistently over the years, nutcrackers do appear to eat the same level of nitrogen values, meaning the same, like if they're eating conifers, they continue to eat conifers, and if they're eating insects, they continue to eat insects during their corresponding seasons that they were consuming it. In terms of carbon, however, we did see a pretty significant decline. And you can see it at the 1920s mark, which could indicate diet shift, though not between trophic levels. This diet shift might be changing between the various conifer species that they are eating. I'm still looking further into it. In terms of spatial scale changes, I have not finished analyzing that part of my data yet, but there appears to be some spatial scale changes in carbon, but not nitrogen. And that is all I have gotten to so far in terms of my data. I'd really like to acknowledge my committee members, the biology department, and everyone that's helped me on this project. You guys have any questions? Yes. So I collected whole feathers, and I actually did find literature that said that the tip of the feather to the base of the feather were the same isotopic signature. So I wasn't too concerned about only taking a portion of the feather, but in terms of taking the feather, I weighed it, and I needed my feather samples to be between 0.5 and 1.5 milligrams for the isotopic analysis. So if it was greater than that, I would trim it so that it was within that range.
In terms of spatial scale, it's mainly latitudinal changes with carbon. And that is kind of expected, as I found some literature on that. But there is an increase in carbon with decreasing latitude. So higher carbon down in Utah versus in like Washington area. I'm not entirely sure what it means yet. And my figures have changed a little bit. So I don't have figures for that quite at the moment. That latitudinal shift is has time factored into it. So time separately has a significant effect from space, but they both are significant. Yeah. Yeah, the carbon is actually very complicated, and I've started off with like one thing with carbon and I keep changing it. Carbon has elevational shifts, it has water stress shifts, it has like spatial shifts over latitude. So I'm still analyzing it and I'm not entirely sure what it is. But the latitudinal shifts do seem to correspond to precipitation at least a little bit. So it's possible that some of the shifts that we see may be due to like climate change factors with precipitation. It was introduced from Europe in the 1920s through, I believe, Eastern white pine. They were brought over from nurseries in Europe. And so when they were planted here, they weren't, I guess, properly checked for invasive species in the 1920s. And so that's how it got started. And it started spreading through different white pine species. White bark pine is one of them. And I believe it got into white bark pine in 1926 specifically from Eastern white pine.
Thank you. Hi, my name is Hunter Korf, and I'm excited to share with you today my contributions over the past 2 years to infectious disease research in leishmaniasis. The title of my project is Bicyclic Lactams Have Leishmanicidal Activity Against Leishmania Major, the Agent of Human Cutaneous Leishmaniasis. Pictured on this slide, we have the promastigote form of the parasite, the sandfly vector, which transmits the disease, and one of the symptoms of cutaneous leishmaniasis. And this bottom photo was taken from a patient experiencing skin lesions by Dr. Blaise Dondji while conducting fieldwork in Cameroon. Leishmaniasis is a neglected tropical disease that impacts 88 countries with 350 million people at risk and roughly 12 million patients. While there are over 20 species of the parasite and 3 clinical manifestations of the disease, Leishmania major or L major and cutaneous leishmaniasis are the focuses of my project. There is an identified need for novel therapeutics in the treatment of leishmaniasis because current treatment options are expensive and toxic to patients. In my efforts, I screened a total of 84 compounds in the Bicyclic Lactam family that were synthesized by the Bang Lab group here in chemistry department. Like many other parasites, Leishmania do follow a life cycle. We can see here as the infected sandfly passes along the promastigote form of the parasite to a novel animal host. There, the immune cells like the macrophage intake the pathogen and try to fight the infection. It is within the macrophage that the parasites transform and multiply, overloading our immune cells and successfully evading our immune system to increase infection. As I mentioned earlier, there are 3 clinical manifestations of the disease, including visceral leishmaniasis, mucocutaneous leishmaniasis, and what we're focused on today, which is cutaneous leishmaniasis. This map of endemicity of cutaneous leishmaniasis shows us that the parasite lives in tropical warm areas where its sandfly vector thrives. Also of note, leishmaniasis is an infectious disease which weakens the immune system, and co-infection with HIV has been reported from 35 countries and is an important consideration to take when treating patients. While this map focuses on cutaneous leishmaniasis, I would also like to share the results from a survey done on visceral leishmaniasis in dogs across the United States in 2001 to 2002. What was so fascinating about that study was there was one positive case in Washington, and that dog was from right here in Ellensburg, which is just what we want to hear, that there are parasites in our backyard. Amphotericin B is the current treatment, the only current treatment that is FDA approved in the United States. Protozoan infections are incredibly difficult to treat because their metabolic processes are so similar to our own. Because of this, AMP-B is toxic to patients and kills parasitic and mammalian cells indiscriminately. Also want to make note here that this is a rather large and complex molecule, which is going to make it harder to synthesize and raise the cost of the drug for patients. While I was screening 84 compounds in the Bicyclic Lactam family, my goals for this project were to identify compounds that were active against Leishmania, that were non-toxic or less toxic, and were affordable and or easier to manufacture. As I take you through the protocol that I followed, we'll be focusing on representative data sets following one active compound that I worked with. To determine the activity level of the compounds, I began with an initial screening assay. Setting up a 96 well plate in triplicates, I included 1 million parasites per well and a treatment at a concentration of 100 micrograms per milliliter. I also added the dye Alamar Blue to every single well. Alamar Blue indicates cell viability because it contains the compound resazurin, which in the presence of mitochondrial enzymes transforms into the pinker or redder colored resarufin. We can see here in this example assay that I ran last year, these 3 wells contain DMSO, are negative control, and have a higher concentration of parasites. And these 3 wells contain amphotericin B and a lower concentration of parasites, with the compounds up above ranging somewhere in between. This is one representative graph showing the activity levels of 12 compounds. And the easiest way to read this graph is that the height of the bar corresponds to the concentration of parasites in the wells. So the higher the bar, the more parasites present. Looking at our compound of focus, 12106AF3, we can see that it's lower than AMP-B, indicating that it was an active compound against the parasite. Also of note here is compound 12108A, which actually increased parasitic growth because it's about 2 times the size of DMSO. Out of the 84 compounds that I screened, a total of 12 came back as active, and another 8 were found to be semi-active. The next important step in drug discovery was to conduct a dose-dependent analysis, which is going to tell us the lowest concentration that we can treat patients and still expect to see an effectiveness from the compound. This followed the same assay protocol as before. I just expanded the concentrations of compound added to the wells. Looking here, I'd like to draw your attention to AMP-B, which is this dark blue line, as well as DMSO, which is this darker red line. And based on this graph, we can tell that the most effective concentration of this compound was 100 micrograms per milliliter. Also of note here is that the 50, 25, and 10 microliter per milliliter concentrations also exhibited activity at the 48-hour mark. An important in-between step from going to in vitro testing and moving into animal models is to determine the cytotoxicity of the cells. One method we can use to do this is flow cytometry, with which I conducted an initial screening of 2 of the compounds to determine the cytotoxicity. Mice splenocytes were prepared and diluted to a final concentration of 1 million cells per well via a trypan blue exclusion. And the treatments for this portion of the protocol were prepared to a final concentration of 200 micrograms per milliliter. I then conducted a dual parameter flow cytometric analysis using the stains annexin 5 and propydium iodine. Annexin 5 binds to phosphatidylserine, which is a phospholipid in the membrane of cells that flips during apoptosis or programmed cell death. Because of this, this allows the machine, the flow cytometer, to differentiate and sort cells based on which stain is present. We can see here, based on this key for understanding the data, we have a large population of necrotic cells for this compound and a smaller population of live cells. Unfortunately, preliminary findings on cytotoxicity were inconclusive because the annexin 5 stain did not properly adhere to the splenocytes. However, the flow cytometric analysis will continue to be used to screen additional compounds in the Dondji lab moving forward. One important aspect of this project is the collaboration between the Dondji lab group in the biology department and the Bang lab group in the chemistry department. Now that I've determined which of my compounds are active and inactive for the set of bicyclic lactams, I can report back to them and we can look at structural similarities and differences that will help guide the decisions on which compounds we screen next in the lab. In conclusion, I'm very excited to share with you the success of my project. I did find 12 active compounds out of the 84 that I tested. And the next steps for these compounds would be moving on to animal trials, and I'm very excited to see where that goes. I'd like to give a very special thank you to the Dr. Bang lab, which synthesized the compounds, my mentors, Dr. Bang and Dr. Dondji, as well as past and current lab students who've helped me, including Cameron Smith, Kira Bush, and Nanju Jian. And thank you so much for coming to my presentation. If you have any questions, I would be more than happy to answer them. Yes. That's a great question. We do kind of look at current drugs on the market to help influence which compounds we choose to test. However, I'm not sure with this set in particular if where that kind of inspiration came from to test those. So. Yes. Mm-hmm. Yeah, so there are 3 forms, and visceral leishmaniasis is the most severe and often deadly when left untreated. Mucocutaneous leishmaniasis focuses on the mucous membrane sites on our body, and cutaneous leishmaniasis impacts the skin. And so it's really important to help treat patients because it's a very forward presenting disease. And so we just want to make sure that we're addressing that. Yes. Yes. Yeah, so the reason we do use the DMSO or dimethyl sulfoxide as our solvent is because it does dissolve all the compounds. So we didn't have any issue. The compounds come to us in a dry form, and then we weigh them out to that concentration and then dilute them in the DMSO. Any other questions?
All right, welcome everyone, and I'm happy to announce our first speaker, who is a researcher in my group, Ruiz.
Hello, everyone. I'm Roy, and thank you for coming for my presentation. Today, I'm just going to share with all of you what I've been doing with Dr. Craig's research lab. So to start off with, the ability of eukaryotic cells to resist deformation against their environmental factors, being able to traffic organelles, and being able to change shape during movement. It depends on the cytoskeleton, and the cytoskeleton is made out of 3 specific filaments, which are actin filaments, intermediate filaments, and microtubules. And for this presentation, we will be focusing on microtubules, the importance of microtubules in neurons. And a neuron is composed of 2 specific components, an axon and dendrites. And this red dot represents the cell body of a neuron, and this other end represents the axonal terminal. And the reason why microtubules are important for neurons is because they maintain the structure of axons and dendrites. They're also important because they also develop a uniform polarity pattern, which helps organize the traffic of organelles, and that's what these figures represent. So usually microtubules that reside inside the axon have a plus end out orientation, which means that these microtubules like to move to the right side, and that's what we represent as a uniform polarity pattern. And on the other hand, microtubules that reside in the dendrites have a mixed polarity pattern, where they have like microtubules that like to move to the right side and the left side, which is not very uniform. And the reason why this is important is because microtubules that have a uniform polarity pattern help make sure that the polarity is not corrupted, because if the uniform polarity pattern is corrupted, then it starts developing neurodegenerative diseases such as Alzheimer's diseases, and that's what we want to prevent happening, and that is the focus of our study. And some examples of corruption that happen is when microtubules are cleaved, flipped, locally nucleated, broken, or bended. And in our computational model, we have included the cleaved and flip mechanism, and we want to see how that is impacted in the model. And microtubules in the axon are pretty much inside. Microtubules are pretty much inside the axon. The axon is like a empty cylinder, and these microtubules are just like floating in them. And microtubules act as the track for organelles to move along the axon. However, this motor protein called molecular motor is what actually transport the organelles throughout the axon by using, let's see, I can't play the video. Well, this protein would just, these 2 blue blobs would be moving on the microtubule, and these green sites would be the spots that these proteins would be attaching, and these blobs would be moving subsequently throughout the microtubule as long as they meet to their destination. And similar process occurs. Oh, now it's playing. Yeah, I'll just show it real quick. So yeah. So, and afterwards, a similar process occurs when 2 microtubules are attached to each other. So let's say one microtubule is attached to the cargo domain and another microtubule is attached to the motor protein domains. And this protein that we're seeing here with the orange dots, this represents the dining molecular protein, and these dining molecular proteins like to move to the left side. And since they like to move to the left side, this causes a sliding force, and this sliding force causes this top microtubule to move to the right side. And that's the same scenario that we see here at the bottom. And the reason why this occurs is because of, according to the third law of Newton, whenever two bodies interact with each other, an equal and opposite force occurs. And the objective of this project is to test how a specific protein, cross-linker protein, affects the polarity pattern, the uniform polarity pattern in the axon, because the cross-linker protein is the purpose of the cross-linker protein is to prevent sliding force to occur, and we will be testing two specific cross-linkers, anti-parallel and parallel, and this system will also contain both of the molecular proteins, adenine and kinesin. So in the computational model, also simulates the two specific populations, one that has the plus end out microtubules and the minus end out microtubules, and the plus end out are the ones that move to the right side, and the other one and the minus end out are the ones that like to move to the left side of the axon. And this bar graph represents the distribution or the population of microtubules of the right side, and the orange bar graphs represent the ones moving to the left side. And yeah, and this second bar graphs represent a specific polarity distribution, and it's the polarity distribution of the microtubules moving to the left side. And proximal, what proximal means is like microtubules that are residing closer to the cell body region, and the distal region represents microtubules closer to the terminal axon. And this is the initial polarity distribution and the final polarity distribution. And this figure represents the movement of the microtubules. So these horizontal lines that we see here, those are microtubules that have not moved at all throughout the first 100 seconds. And this purple line represents microtubules that are moving really fast continuously. And this sporadic pattern that we see are a microtubule that has been moving back and forth, which is very normal because we have two specific motor proteins, a kinesin and a dining. And since kinesin likes to move to the right side and dining likes to move to the left side while walking on a microtubule, it creates a tug of war. And that's why we see this this pattern. And we'll just skip that. And in case one, so what I did here was I checked for the polarity distribution against an effectiveness with kinesin, motor kinesin. And what we saw is that we have a lot of microtubules residing, or microtubules that like to move to the left side in the proximal region where they're close by to the cell body. And after these microtubules have done the interactions with the cross-linkers and molecular proteins, we noticed that most of them left the axon, but some of them still reside in the distal, which is what we want to see because usually whenever we have a kinesin motor protein, these usually like to move to the right side, which is closer to the axonal terminal. And afterwards, I did a case with polarity distribution with dining molecular proteins, and we saw that we had a fair distribution in all the regions, but afterwards we saw that most of the microtubules moved to the proximal region, which is what we want to see because because the molecular motor proteins like to move to the left side. And afterwards, I tested another polarity distribution where we have parallel cross-linkers, and we saw that the parallel cross-linkers actually assisted the dynein molecular motor protein, which is an indication that all of the minus and out, or all of the microtubules that move to the left side, are moving to the cell body, and that's our goal. We want to move into the cell body because if they move to the cell body, they're able to readjust and be able to function how they're supposed to function in the axon, and to help form a uniform polarity pattern. And then afterwards, I tested a cross-linker that has a preference with anti-parallel microtubules, and we did not see any change, and that is fine, because that means that the sliding force was not very effective in this system. Yeah, and so final remarks in the project is that the crosslinker that is parallel is much more effective in order to create a uniform polarity pattern, and also an anti-parallel crosslinker is also helpful. And the medical relevance in this project is that it's helping us understand a bit better how microtubules are behaving in the axon, and how we can better approach, or how to study how neurodegenerative diseases are developed. So yeah, thank you guys. Yeah, so Kaon represents the effectiveness of kinesin attachment. Yeah, and it means that that we have more kinesin proteins in the system, and the other one just represents that we have more dynein proteins in the system. Yeah, so it's simulation data, but we're using experimental parameters because we're using so yeah, it is a simulation, but we're using experimental parameters that have been used in other tests and other research projects.
Okay. I'm not sure how, but I don't know if you can find the way to turn the AC off, that might be a solution too. Hey, Nick, do you want to come take a glance here and see if you can see which one's yours here? All right. That it? Yep. Perfect. Okay, let's get you into slide share mode. And I'll go ahead and. All right, so next up we have Nick Cameron from the PHX lab.
Hello, I am Nick Cameron. I did my undergraduate research here at Central Washington University with Andrew Piacek, and I did my research on investigating a non-invasive method for monitoring intracranial pressure change using sheep stools. The reason we were doing this is because currently there is no medical way that is non-invasive of measuring ICP or intracranial pressure. And we wanted to develop a way that we could possibly use the resonance of a skull to measure frequency changes which correspond to the intracranial pressure. We used a sheep head because it was one of the best access we had to that would correspond to a human skull. And we also investigated the sensitivity of skull vibration response or skull vibrational response to environmental factors, and how it would correspond to previous experiments done with a metal aluminum shell. Some background, some students here had previously done research using a metal sphere or a aluminum shell that was filled with fluid and increased pressures from around 0 to 100 psi. From that data, they found that there was a corresponding change in resonance frequencies to the corresponding pressures. And after that, they developed a COMSOL model to model the aluminum shell, which was consistent with the frequency shifts were consistent with the measured values that they had previously done. And then later on, a student came along and studied small pressure changes using a modal hammer, and consistently found that even for small pressure changes, we were still seeing resonance frequency shifts, which led me to why I am studying what I am. Here is the experiment that they worked with. They had the metal sphere, which can be seen on right around here, and using the pressure or force sensor gauge on the, or force sensor hammer on the bottom left, and the pressure gauge on the top left. From that, they found that for higher frequencies on the top left, around 9.2 kilohertz, they were seeing relatively large frequency changes, around 0.2 hertz per PSI. There were some resonance peaks that were experiencing no frequency changes, and for relatively low resonance peaks, they were seeing negative frequency changes. On the very bottom, we have a linear graph of the low pressure data that shows that even for 0 to 2 psi, we were still seeing around a 0.7 hertz per psi change. This led me to what I began working on with the ovine head test setup. Here I have one of my 3 setups that I began working with, and that includes the test subject resting on the table and it being clamped via support. I also, in this picture, I have a mechanical oscillator that is being driven with a swept sine signal, which a swept sine signal is a driven oscillator that ranges from frequencies of 3 to 22,000, or as best Dr. Piacek puts it, it's a signal that goes, and we also took data with a impulse hammer, or impulse force hammer, which will be shown right here. As you can see, we had about a 12 millisecond window for this accelerometer data, which is very short when you're trying to take Fourier transforms of it. In comparison, the metal sphere was ranging with about 0.6 of a second, and much like noisier, so we determined that the damping in this system must be very high, and it will be a large issue that we will have to work with. The graph on the right is of this accelerometer data from the sheep skull. Each different trial corresponds to 1 tap of the sheep skull. And in the data, the accelerometer stays the exact same, and we are striking the hammer in the exact same location. This showed us that that all of these resonance peaks above 1.5 thousand would be completely useless because none of them correspond to each other. And we chalked it up to being signal processing. So we decided to move on to the impulse driver, or the mechanical oscillator, with a range of 3 to 22,000. On the left, we can see a much cleaner and louder signal with 3 to 4 relatively high peaks that correspond to some resonant peak or possible resonant peaks. On the right is the different driving locations that we studied as well, and how the accelerometer was attached to the skull. And this was some of my initial data taken on the ovine skull. As you can, the only difference in this data, we have the exact same accelerometer location. The only change was removing the driver itself and trying my best to replace the driver in the exact same location. From this, we determined that having it resting on the table would not be viable, as we were seeing insanely large amplitude shifts as well as frequency shifts. Moving on, we tested out the clamped jaw, and we were still seeing large amplitude shifts and large frequency shifts. One thing to notice that we were seeing resonance frequencies at around 8,000 hertz for both of these, and at around 16,000, which was much better than what we were getting from the modal hammer. And for the final, when we suspended the head, we were getting much more clean data. We still had slight frequency shifts, but the amplitudes themselves were not shifting about half the size. We were getting very consistently very consistently the same amplitudes. And finally, we took some data with different locations, which confirmed our previous beliefs in that driving near certain antinodes will yield larger amplitudes, and that and consistently location C was greater than location B, and consistently location B was greater than location A for multiple different trials. So this confirms that we are getting relatively alike data, but we still are seeing small shifts that could be very hard to recognize when we're looking for shifts less than 0.7 of a hertz when we increase by 1 psi. And in conclusion, we determined that the mechanical driver with the swept sine signal was the most reliable for taking data, even though it is unpreferred when taking data for resonant frequencies. We also determined that suspending the head on strings was the most reliable in response measurements, rather than clamping it on the table or resting on the table. We also concluded that driver contact location affects amplitude and resonant peaks, and that even just slight changes could impact the hertz or impact the frequency significantly. And we also are still addressing the problem with large damping, as animal model provides challenges in obtaining these reliable frequency responses, and it could be relatively difficult when looking at small intracranial pressure changes. And in the future, we hope that we will be able to get more consistent and reliable frequency plots, and then in the future, hopefully be able to change the pressure inside of the skull and use a possible swept sine signal to look at possible frequency shifts. And thank you. Are there any questions? Yes. Well, relatively, the sheep head itself has a very small area. If I can go back to that. We were trying to focus most of our time in this area because it is the closest to the actual brain inside of the sheep skull. which we realized a sheep stool later on might not be the best of animals because it has such a small area that we were trying to analyze. And we wanted something that would mimic the crane or like a human stool as best as possible. In the short answer, we would have to reverse engineer basically what we're doing here. That would take a lot more research and a lot more time to understand. I think ideally we would first want to focus on a cadaver that we could control to test and see if we actually can see these changes in frequency. Thank you.